all of the talks that we've had in the last couple of days, some incredible detailed analyses from coral reefs and other habitats, and now Frederick's very critical and detailed assessment of aquaculture and bivalves and oysters. I'm going to talk about our attempts to start to develop, that's why I put towards a blue carbon model, because we're nowhere near what previous speakers have talked about. And I'm looking today at the European flat oyster, Ostrea edulis, as opposed to the Pacific oyster in the aquaculture industry. And of course, in France, we still have a very healthy, wild, semi-wild flat oyster cultivation process that Frederick just mentioned. So I'm going to be talking today. My name's Bill Sanderson. I'm from Heriot-Watt University. I've spent half my career in the university system, and the other half as a government scientist, nature conservation side, working on the applied science and monitoring needs of habitats and species of high biodiversity conservation importance. And today I'm going to speak quite a lot about work that's been done by Hannah Lee, who was in my team for a few years, and these other people here have contributed as well. And my major contributors in this particular project are the Glenmorangie Company, Blue Carbon Forum, through the MASTS, Marine Alliance for Science and Technology in Scotland, and Napier University as well. And some of this work was done at St Abbs Marine Station when I was the co-director of research there. There we go. There's Hannah Lee. That's a lot of the data I'll be presenting was hers. Now, the first point I want to make is some of you will have seen this before, others may not. This is from Beck et al. In 2009. It was by the Nature Conservancy. We did a global analysis and showed that oyster habitats are functionally extinct around the world. We've lost historically over 85% of them, and perhaps in a European context even more than that. So this is kind of like my starting point, is that this kind of gets in the way of carbon studies because there aren't very many of them. So finding these habitats is quite a headache. And also I'd like to point out this piscatorial atlas from the 1880s by Olsen, who shows these oyster habitats on the left panel here over extensive areas around our coast and into the Manche or the English Channel, and the Dutch and German Bodensee out past Helgoland, the Dogger Bank. So 200-mile stretches of abundant oyster habitats. In the further forth, where I'm based near Edinburgh, one of these little rings on the map, we're talking about an oyster bed that was 20 miles long by 6 miles wide, about 300 square kilometres. So we're talking about oyster habitats that have disappeared from our coastal waters by the start of the 1900s, and were fished. We've ate them all, basically. We've fished them to extinction in most places. And for those of you who may be less familiar with this story, you might be thinking that Olsen in the 1800s might have gone a bit crazy with his crayons here on the map, and maybe this is very realistic, but some excellent historical ecology by Ruth Thurston and many other workers recently published has picked out lots and lots of records of positions and extents of oyster beds throughout the Northeast Atlantic and throughout Europe. And these kind of like these records, which of course aren't the whole story, mirror to some extent the extents that Olsen recorded from his records from the fishing industry. Now, the other thing that's quite curious is that when you assess all of this information together, Feline Zeromicson and others have recently published another paper using the IUCN criteria and describe oyster reefs in the European context as a collapsed ecosystem. We basically have them only as scrappy remnants of what they were in the past. So we're talking about a habitat that's a bit hard to find, and this means that over the last 10 years there's been quite a rapid increase in the number of projects that have started to try and restore oyster populations. And when we're Feline Zurumgason and myself have just had a paper accepted on this concept of restoring ecosystems at scale, and we've put this recent collation, the number of oyster restoration sites in there. So you can see things are accelerating. We have more than 50 sites now throughout Europe where people are starting to restore oysters. And all of this has started to happen within the last 10 years. And in that time, the UN Decade on Ecosystem Restoration has come along, trying to put rocket boosters on the concept of restoration in the terrestrial and marine environment. And also the EU Restoration Law has come along, which is starting to attribute targets to some of these things as well. And member states are now going to be thinking about what they can restore on land and seas. So this is a growing area, and we haven't got particularly good reference sites. The projects that I've been very closely involved in, this is the Dornoch Environmental Enhancement Project, which is going for 10 years now, as one of the early adopters of oyster restoration in the European context. And we've been through lots of different stages with this project. We've investigated the environmental history of the site to establish whether oysters were there. We've done survival trials to find out whether the oysters would still survive in the present day conditions. And then we've started to recreate the habitats and optimize that process of restoration. And we're, like many people now, trying to move these restoration projects to scale. Also in the Firth of Forth, that very large oyster bed that I talked about earlier in Olsen's Piscatorial Atlas, there was this huge oyster bed. And during the 1800s, we have fisheries records of 30 million a year being extracted from this bed over a period of at least 10 years. So the scale of extraction was eye-watering, and this restoration project has a huge hole to try and fill. We're 4 years into this now. It's quite a large community restoration project involving a lot of different community engagement partners. We intend to influence the policy behind the restoration process. And we've put 40,000 oysters in the water here so far, which is a small drop in the ocean, if you don't mind the pun. Now, the reason we might want to restore oysters is why I get into this. I come at it from a biodiversity conservation point of view and a restoration of ecosystems. But the reason behind why people would invest in restoration is generally related to the ecosystem services, which I sometimes regard as the business models for restoration. So oysters form these complex habitats on the seabed with the shell material, which creates lots of niches for other species. Biodiversity increases, and also filter feeders. So in the process of going about their business, they're also filtering the water and taking the particulate matter out of it, which can result in denitrification, soaking up some of the nutrients in the system, increasing the habitat complexity, and therefore perhaps increasing fish production, as we've seen in other oyster beds elsewhere in the world, improving water clarity, stabilizing sediments, etc. So these are the kind of business models for restoration. And I suppose I became interested in carbon because I was becoming a bit alarmed by some of the rather glib stories that I saw in the press about restoring oysters to offset climate change. And this is what motivated me to get into looking at this very carefully, well, as carefully as I could. So I share some of Frederick's perhaps sceptical views here. And the other thing to appreciate is that the benefits of oyster restoration are actually quite poorly understood. This assessment by Feline Zurumgason and myself and others in 2020, we looked at what was known about the different species that were the focus of restoration efforts around the world. And for Ostrea edulis, there was an awful lot of question marks against some of the provisioning, regulating, and cultural services. And the ones ringed in yellow here are the ones that my team has done some work on. And the one in purple here, the carbon story, was one we decided we needed to try and find out something about. And a lot of people, I think, are guilty of looking at these habitat types. This is, by the way, an oyster habitat on the southwest of Scotland, which I'll talk about later as well. And people are guilty perhaps of looking at these habitats and saying, hey, look at all that calcium carbonate, therefore that must be a way of fixing climate change because that calcium carbonate is going to be quite persistent. And indeed, in some of our earlier studies, we were able to find shell material that was 6, 8, even 8,000 years old. You know, when it gets buried in the sediment, it can be very persistent and becomes subfossil and then come back up again. So this material is around for a long time. I think some people believe by looking at these habitats that they must be carbon mitigation story, climate mitigation story. And that's the point I just made. And these pictures come from Naomi Kennan's work in Loch Ryan. And this is where I kind of take a look sideways, because before I got into oyster restoration 10 years ago, my first love, I suppose, was horse mussel habitats, which I've been looking at since the 1990s. And these are things that we haven't targeted with fisheries. So we still have intact systems, horse mussel systems, albeit some of them have been quite badly damaged, incidentally. And these horse mussels are about the size of your hand, modulus. They're not mitulus, they're quite big, and they're quite long-lived. And this particular habitat that we looked at back then is about 375 hectares. And in that area, you can find mussel clumps which equate to about 600 per square metre. It's incredibly dense. And the thing that I found fascinating was that back then we could see in the sub-bottom profiling, this image on the right-hand side, the underlying lag gravel deposits from the last glaciation. And you can see, therefore, that the mussel muds and the shell material has been built up by 1.5 meters, even 2 meters in some places, above that reference point. And you get this characteristic undulating seabed type. So I look at these habitats, and they're aggregative, bivalves that form these habitats in the subtidal. And I increasingly believe that oysters are the sort of southern analogue of these sort of northern, bivalve habitats, and they occur in many of the same places historically. And back more recently, Flora Kent did some work on deposition in horse mussel habitats, and she took a look at this particular horse mussel bed and calculated there was 1.87 million cubic meters of this mussel mud, which is odd in that it's a tide-swept area, but it's got a high mud content and lots of shell material, which is held down by this living skin of these mussels growing over the top. Okay, so we started looking at oyster habitats, and rather like we heard from Nick and others yesterday, you can look at these things at different scales. You can look at the organism level, you can look at populations of oysters, you can look at the actual ecosystem or the habitat type, and you can look at the wider exchange of carbon between these habitats and others in the system. But we were pretty stumped in that we can't really study wild oyster habitats because there just aren't any of them left in any great numbers when we started the study. So we were focusing in on the organismal level. And of course, if you're thinking about the oysters themselves, as I mentioned earlier, a lot of people get preoccupied with the calcification of the oysters when they don't look into things when they look at things very superficially. But of course, they're respiring whilst they're living their lives. And the thing that I was quite aware of from looking at the horse mussel habitats was this biodeposition, the fact that shellfish bundle up particles and they poop them out onto the seabed, whether it's feces or pseudofeces. And we took these three main components of the budget for carbon in oysters to look at first. So, of course, we've been putting all these 2-year-old oysters into our restoration sites for some time now. So we were able to look at the annual calcification rate by recovering oysters from our restoration site in the Dornoch, which is in the northeast of Scotland. We took samples of these oysters at the time of deployment, and we recovered them in subsequent years, every time in September. So for 4 consecutive years, we were able to look at the growth rate of these oysters in terms of their dry shell weight. And we were able to look at the carbon content of that increase in shell by acid digestion. And this is what they look like, just eyeballing them. So they obviously increase in size over the time at the restoration site. And you'll see that they, in terms of their linear length, they slow down, but their shell gets thicker. And then we also looked at the respiration rates. So we used incubation chambers a lot like what was just being described a minute ago. We had 6-hour incubations. There were 900 ml containers. We had an oyster in some, and then we had controls in others. And we monitored all the same kind of things that we just heard about in the Q&A in the last talk. And to cut a long story short, we repeated this a couple of times, and the samples were analysed for their total alkalinity and DIC. We also looked at deposition, and we used these shallow containers with basically scouring pad material on the bottom to capture the poop that the oysters produce. And we used ambient seawater flowing through the aquarium system. It's ABS, it was an open aquarium, taking its water directly from the sea. And we had 3 treatments in this because we were interested in the live oysters and the dead oysters, because there might be a passive deposition effect from the topography of the oysters, the shape of them causing turbulence and the dropout of particles. We have control as well, of course. And from that, we were able to look at the total particulate matter that's still out in these containers. And from that, we were able to look at from the loss of ignition, the total organic matter, the total inorganic matter. And by checking samples using a CHN elemental analyzer, we were able to work out the carbon percentage carbon in those segments as well. So to cut a long story short, the shell length obviously increases, but tapers off, as I said, but the shell weight continues to grow over the time that we have them in the restoration sites. So we're really looking at the first part of the restoration process here, when the animals are relatively young. And we can see there that the overall, when you get down to it, they're putting down about 1.4 grams of carbon per year. And they're producing, if you work out the release of carbon from the calcification process as well, it's about 0.8 grams of carbon per year through that route as well. And from the respiration work, we're able to work out that the oysters are producing about 1.7 grams of carbon per year. And we can work out from the depositional experiments that there's a trebling of the deposition onto the seabed through the active feeding of the live oysters compared to the control of the dead oysters. And we didn't see in this experiment a passive depositional effect from the turbulence created by the shell material. So basically you can work out the organic and inorganic carbon content that's settled out from the feeding process. If we put all that together and we look at just these 3 components, which I fully admit is an incomplete picture, we end up, and of course I have to acknowledge that we did our respiratory experiments and our depositional experiments in the early part of the summer before the bloom. So basically, this is not a complete picture. But in this experiment, in this piece of work, we find a carbon budget of 1.5 grams of carbon per oyster per year going onto the seabed. And of course, when we look at this framework, we're only operating at this organismal level. We also have to think about what happens with wider populations, and also the overall ecosystem and the exchange with other habitat types. And if you look at the organismal level, we've clearly got a lot more to do with temporal and spatial variability, and oysters at different ages, and between different sites as well, because of course they're not all going to perform differently in different places. And then also when you look at the population level, there's a lot more to do with perhaps synergistic effects when you've got a lot of filter feeders all doing their thing at the same time, but also when they're creating physical structures and clumps on the seabed, and there's quite likely to be some passive depositional effects and entrainment of organic material into oyster beds. And we heard Nick mentioning that kind of thing yesterday. And I've seen, detrital algae and seagrass material sort of settling out in oyster beds and dying and falling into the cracks there. So there's clearly work to be done there. And as we heard from Sergio yesterday as well, it's going to be a much more enhanced picture when you talk about the whole community. Here we have hydroids in the left growing on oysters in the wild, and we have sponge material, and we have anemones, and all sorts of other things, as well as red algal turfs growing on top of the mass of oysters. And those pictures will be in the paper that's coming out, so it's probably in Respiration Ecology that we just had accepted. Now, we should also expect quite a bit of variation between sites, and you'll see that I go look sideways again at this piece of work we did with Jose Frenius Franco leading, where we looked at horse mussel beds from Norway all the way through to the Irish Sea throughout Scotland. And we're able to see that with increasing densities of horse mussels, we get obviously an increase in the biodiversity and the organisms associated with that. And we also get a massive increase in the biodiversity when we come into the shallow water, where we have algal communities, where you've got primary production going on as well. So the density of oysters will vary in different situations, in different restoration sites. We'll get lots of different associated organisms, which will all contribute in different ways, I think, to the overall carbon budget for oyster habitats. And of course, when we look beyond the habitat and we start thinking about things at the ecosystem scale, the amount of carbon that's going into these habitats is likely to change when they are connected at an ecosystem scale with other things. So, maybe might be where fragments of algae end up in oyster habitats. Maybe the fragments of seagrass habitats end up in oyster habitats. And they trap the seagrass and algal fragments and build up carbon deposits that might be akin to the horse mussel carbon deposits that we see on the Flint Peninsula in the Irish Sea and the work I mentioned earlier. There's quite a lot more to do on the ecosystem services flow between habitats. But of course, that's difficult when most of those habitats aren't there anymore. But I'm pleased to say that we do now have, in the last couple of years, we discovered fairly unimpacted oyster habitats that we believe have never been fished. And they're the ones that I'm going to next. So coming back to this business model concept, we started doing this work because, of course, when you mention carbon, people's ears break up and they get excited about it. And we were very keen with the work that we've been doing in the Dornoch Firth project, the DEEP project, that all of our claims were made on sound ground. And overall, a fully restored oyster habitat in the Dornoch Firth, which would be 4 million oysters over a 40-hectare area, might be expected over a year to draw down about 9 tons of carbon onto the seabed using the figures that we've got. And this is actually quite a moderate number compared to some of the more traditional blue carbon habitats that we were hearing about yesterday. This, of course, is only our first understanding of the scale of the carbon flux. We don't consider it to be a big deal, although, as Nick was pointing out yesterday, the real value in these sort of habitats is the creation of the carbon stores, not necessarily in the amount of net carbon deposition that goes on. So, moving ahead, and considering the functional understanding of oyster habitats, I'd say this highlights really the sort of real importance of considering the functional habitat, the functional role of these habitats in a management context. We're much more preoccupied now with the biodiversity gains that you get from oyster habitats. We're able to establish that you get about doubling of diversity indices over a decade in a potential restoration context. And we think that the water filtration value of the oysters themselves is perhaps more valuable than the overall carbon going onto the seabed. But nevertheless, the creation of carbon stores is nevertheless important. And we will always be in a situation, I think, in the restoration world where investors will want to know what they're getting for their money. And at the moment, they get quite a lot of biodiversity gain. They may get some good water quality improvement, and they'll get a moderate amount of carbon deposition. And from a conservation management point of view, of course, these sort of shellfish habitats are the focus of marine protected areas and conservation management. So the importance of these carbon stores is underlined in that kind of context. And also from a wider marine spatial planning context, the Marine Strategy Framework Directive, the Marine Spatial Planning Directive, all seek to maintain good conditions, sustainable conditions, and maintaining and encouraging carbon stocks is perhaps something that we might see more and more through those sort of routes. So I'll wrap up there a little bit early, but just to say thank you for listening. These are all the contributors and partners that have been involved in these two major restoration projects that we've got on the go. And so far, we've managed to get 157,000 young adult oysters back into the marine environment as of our last deployment, which was yesterday. And I'll stop there. Thank you very much.